These are the lecture notes for slide set 2B, chapter 2B, and we're starting with slide number 12, okay? We had left off with the definition of acids and bases. So acids are going to have a high concentration of hydrogen ions or protons, and bases are going to have a lower concentration of hydrogen ions or protons in a solution. So the pH scale measures the concentration of hydrogen ions. The lower the pH number, then the greater the concentration of hydrogen ions. So pH ranges from 0 to 14, and acids are less than 7, bases are greater than 7, and then 7 is neutral. If you increase the pH of a solution from 5 to 6, that's going to be a 10 times less acidic. Or you could say that like if you read the statement down at the bottom, the pH 5 solution is 10 times more acidic than the pH 6 solution. Or you could say pH 6 is 10 times less acidic than pH 5. pH 5 is 10 times more acidic than pH 6. So if you go up again, it would be like pH 7. Then you could say pH 5 is 100 times more acidic than pH 7. So every time you go down in a pH, you multiply by 10 times more concentrated in hydrogen ions, or 10 times more acidic. All right, so acid is less than 7, neutral is equal to 7, distilled water is neutral, so the pH of pure distilled water should be 7. And then alkaline solutions. Alkaline is just another term for basic, and those pHs are going to be higher than 7. And the concentration of hydrogen ions would be low. This is a illustration that shows some common solutions and their pHs. So starting with very, very basic, a strong base is sodium hydroxide. Oven cleaner, which contains lye, or oven cleaner and lye, which a lot of times both of these contain sodium hydroxide, are going to be close to a concentrated solution of sodium hydroxide. Ammonia, bleach, these are strong cleaners, and they tend to be very strong bases. Now, egg whites have a basic pH. Blood has a pH close to neutral, but it's going to be slightly basic, 7.4. And then milk is slightly acidic, but it's a weak acid. It's really, really close to neutral. Then black coffee is pH 5, so coffee is slightly acidic too. Wine is a stronger acid. Lemon juice and fruit juice are stronger acids, and vinegar. And then hydrochloric acid or stomach acid is a strong acid. Neutralization reactions are when acids and bases are mixed together. If both of those have the same strength, then they're going to completely neutralize each other. So here we have sodium hydroxide plus hydrochloric acid. When they are mixed together in solution, then at the same concentration and the same strength, then they're going to neutralize each other, and they will form sodium chloride and water, which is salt and water, which is a neutral solution, pH of pH 7. Okay, a buffer is any chemical or group of chemicals that resist changes in pH. The most common buffer system in the human body, sorry, I see where there's a little mistake here too. I don't know how to fix that, but okay, well, at least you can see here the reaction. the reaction, just imagine an arrow in this middle section. I'm not sure what the problem is, but this is the carbonic acid bicarbonate buffer system. Basically, carbonic acid forms when carbon dioxide and water combine. So carbonic acid can, if the arrow points toward the right, it can break down into bicarbonate and hydrogen ions. And this will happen if the pH of our body fluids rises, because the increase in hydrogen ions will lower the pH. Now, if the pH lowers, the pH drops, then the reaction will go in the reverse direction. Hydrogen ions will combine with bicarbonate ions, and then the arrow will point toward the left, and carbonic acid will form. And this will remove hydrogen ions from the solution, and it will raise the pH. So buffers resist changes in pH, and the carbonic acid bicarbonate buffer system is a very important buffer system in our blood. Now, this is a note on homeostatic imbalance 2.1. Enzymes in the body work within a very narrow pH range. So if the pH in our arteries, an arterial pH, is 7.0 during CPR or cardiopulmonary resuscitation, this is going to predict a poor outcome because 7.0 is too low. Patients who present with an arterial pH of less than 6.85 rarely even survive. So the pH needs to be closer to 7.4. Needs to be between 7.35 and 7.45. Okay, so that just lets you know how important pH is within our body fluids. All right, now let's talk about what it means for a molecule to be classified as organic. An organic molecule contains the element carbon, but remember, I think I've already told you this, it's also going to contain hydrogen. If it doesn't, it's not considered organic. So CO2, which is carbon dioxide, and CO, which is carbon monoxide, are inorganic, even though they're gases that contain carbon. And even though living cells produce carbon dioxide, and we produce carbon dioxide in our cells, and then we breathe it out as a waste, it's still inorganic. So organic has to contain carbon and hydrogen together. Carbon will form 4 covalent bonds with other elements, and carbon always shares electrons with other atoms to form compounds. The major organic compounds, there are 4 classes, and they're called carbohydrates, lipids, proteins, and nucleic acids. So many of our organic compounds are very large chains, so we call them polymers. And the building blocks of polymers are called monomers. For example, one of our polymers is a protein, and the monomers or building blocks of proteins are amino acids. When polymers are synthesized, that type of chemical reaction is a dehydration synthesis. And when polymers are broken down, it is hydrolysis. So this is showing you the dehydration synthesis of a polymer. You take 2 monomers, remove water by removing an OH from one monomer and an H from the other one. And water is removed and the 2 monomers are linked together. That is a dehydration synthesis reaction. The example they've shown you here is combining the monomers glucose and fructose, which are monosaccharides, removing water, and producing sucrose, which is a disaccharide, or 2 sugars. And sucrose is also the name for table sugar, by the way. So in the production of sucrose, water is also produced or given off as a product. And that's why we call it dehydration synthesis, because water's removed from the reactants. All right, carbohydrates include sugars and starches, as well as some other carbohydrates found in plants. And glycogen, which is the storage form of glucose that's found in our liver cells and our muscle cells. So carbohydrates include simple sugars and then complex carbs, which are starches and other complex carbohydrates like glycogen. So carbohydrates contain carbon, hydrogen, and oxygen. The ratio of hydrogen and oxygen are 2 to 1. Monosaccharides are the monomers of carbohydrates, and they're also called simple sugars or one sugar. Disaccharides are 2 sugars, and polysaccharides are chains of monosaccharides, and they're the polymers of carbohydrates. So an example of a monosaccharide is glucose, which is a hexose sugar. That means its shape is like a hexagon or 6-sided figure. It also has 6 carbons. And then ribose and deoxyribose are found in nucleic acids. Deoxyribose is the sugar in DNA, and ribose is the sugar in RNA. So they are pentose sugars. They're shaped like a pentagon or 5-sided figure, and they contain 5 carbons. So the monosaccharides are glucose, fructose, and galactose. These are monosaccharides called hexose sugars. And then we have deoxyribose and ribose, which are our pentose sugars. Now, we already talked about the disaccharide sucrose, which is formed from glucose plus fructose. Maltose is a disaccharide formed from glucose plus glucose, so 2 glucose molecules. And lactose is formed from glucose plus galactose. You can see those reactions here. So these are our disaccharides: sucrose, table sugar; maltose, which is the sugar found in malt powder and malt beverages like malt liquor. And lactose is the sugar found in milk. Then we have our polysaccharides, which are long chains of monosaccharides. The important ones are starch, which is found in plants. It's the storage form of glucose in plants, and a lot of times starch is found in plant roots like potatoes. And glycogen is very similar to starch, but it's the storage form of glucose found in animal cells. Glycogen is what forms in our liver cells when our liver cells take glucose from our blood and store it. They store it in long chains called glycogen. So this is showing you an image of a polysaccharide. It looks like starch, chains of glucose molecules, starch or glycogen, either one. And now we're going to go on to lipids. Lipids also contain carbon, hydrogen, and oxygen, but they have a different ratio of carbon, hydrogen, and oxygen. I think it's less oxygen than you have in carbohydrates. Lipids are insoluble in water. They are where carbohydrates are mostly soluble in water because they are polar molecules, most of them. Lipids are nonpolar covalent molecules, so they're insoluble in water. The main types of lipids are triglycerides, which is the name we give to body fat. Phospholipids, which are found in, I don't know why the H is capital, except for, it was probably, here, let me just fix it. It was probably in the, like automatic editing. All right, there we go. That's better. So triglycerides, phospholipids, which are found in plasma membranes of our cells, steroids like testosterone and estrogen, and steroids are formed from cholesterol. So cholesterol is also considered a steroid. And eicosanoids, which we'll talk about in a minute. So our lipids are triglycerides, which are composed of, the 3 is from 3 fatty acids and 1 glycerol. They mainly function in long-term energy storage, insulation, and protection. Some of our organs, like our heart and our kidneys and our brain are surrounded, or our heart and our kidneys especially, are surrounded by a layer of fat to protect them. This is a triglyceride. This animation is found on slide number 31 if you want to go back to the slides and watch it. And we will start here with slide 32, and we will talk about the differences between saturated and unsaturated and trans fats.